All right, guys. And now we're going to start the structure of the proline. Proline, it's one of those tricky actually amino acids. It's not aromatic, but because there's actually a ring in it, that's why some of the people they think this is the aromatic amino acid. But this is not the aromatic amino acid. This is the non-aromatic amino acid, and it doesn't have any aromatic properties. So the great thing about the proline is we're going to start actually draw the structure first, and we're going to talk about the other stuff. So imagine the same thing that we were doing for the other amino acids. We're going to come over here and put the carboxyl group and then connect. And here, when you come here and we try to draw the line and try to actually put the amine group to attach the amine group to it, we're not going to do that. What we're going to do, we start making a pentagonal shape or ring, and we're going to bring it till here. So imagine here our NH2 group has to be attached, okay? But because here the N has to actually bond with another atom on this side, so what's going to happen, the NH2 is going to be converted to NH. So this is proline, very easy. So instead of all those, imagine for example you have the alanine or some other amino acids, it seems like it's been folded to itself. You know what I'm talking about? For example, if you remember the alanine, or for example valine, when you actually were trying to get the structure of the amino acids valine, we're going to come here and put this, and let me just speed up a little bit, NH2, and then here, and I told you we're going to attach the V to it. That's it. This is the valine. Imagine this valine has been kind of like folded into itself. You know, that's actually how the structure of the proline, I define it this way. We're going to make this structure a little bit cleaner in order to understand it better. And one of the reason is we put the H over here because we want to make sure that you guys understand that this N is connected to this ring, not the H. H is connected to the N. Because if you remember electrovalences of the N, it was the last valence electrons, the external valence electron, the one that actually go to the bonding and covalent bonding. It was like one pair of electron right here, and 1 and 2 and 3 free electron that actually make the bonds. So these three, those are the ones that make the bonds. And here we see that two of them actually it's been involved with two of them is going to be involved with the atoms here, like a carbons here and here, and one of them is actually connected to the H. All right, so we're going to make the structure a little bit cleaner in order to understand it better. Okay, we come here and all right, let's make the structure a little bit cleaner. So what we're going to do this time, we're going to start with this ring. So because we want to make it like, imagine when we were in the childhood trying to actually draw a house, we were just putting a ceiling and then coming down like this. All right, and here we're going to put the N and we're going to connect this to it, and the H we're going to put it at the bottom, and then we're going to add the other part of the structure. So this is the perfect proline structure that you see it in picture, this one. So proline, due to the actually what I call this kind of like fold that it takes over here, and due to the small structure, it's so useful in protein folding. That's why the proline actually is used for the protein folding and giving it actually that structure of the folded structure of the proteins. Also, another thing about these proline is, if you remember, I said the proline plus hydroxy proline plus glycine, they will create what? Collagen. This is a very important aspect of the proline and hydroxyproline. Okay, now that we are here, let's draw the structure of the hydroxyproline as well. Now we know how to draw the structure of the proline. Hydroxyproline is going to be so easy. Imagine this is the proline. what the hydroxy tells you. Hydroxy always in science, and especially organic chemistry, is talking about the OH. This is hydroxy or hydroxyl. Okay? So we're going to see, this is the proline, what we need to do. Okay, we're going to draw the structure of the proline over here. OH, come down here. Try to make it nice. And here, and here. Okay, great. The only thing that we need to do in order to make this proline, get hydroxyproline, we have to add the OH here. That's it. This is the hydroxyproline. Hydroxyproline. All right. You see, organic chemistry and the biochemistry, they are not that hard. We have to just pay attention to what we are doing and what we are hearing. So it's very important in the protein folding. And also because of this actually structure that it has, it used as a kink to the folding of the protein. All right, let's go back to our presentations here. And what I have to talk about a little bit with you guys is this thing before we get to the aromatic ones. So these are the essential amino acids. What does it mean by the essential amino acids? Because amino acids, between these 20 proteinogenic amino acids, there are almost like 1, 2, 3, 4, 5, 6, 7, 8, 9 of them. They're going to be essential amino acids. What does it mean by that? Essential amino acids, those are those amino acids that in our body we don't have any pathway or metabolism pathway for them in order to create them. So that means our body doesn't make them and we have to take them from the diet. And that's how actually they become essential amino acids. And too much of it is going to cause a problem for our body, and lack of it or loss of it is going to cause other problems. That we're going to talk about some of these amino acids when we are talking about the clinical correlations of them. You understand that. And these 9 amino acids, they're going to be histidine that we're going to talk about later on. Isoleucine that we talk about it. Leucine, you see, these are the amino acids that you have to get it from the diet. Lysine, methionine, which is one of the important ones that we're going to talk about it after proline. And then we have the phenylalanine, threonine, and tryptophan, valine. Valine, remember the valine, leucine, isoleucine, all there are essential amino acids. That means our body is not going to be able to make them. So that's why we have to get them from the diet. And this is one of the important things that I want to talk about for you guys. Then after that, we have the non-essential amino acids. Non-essential amino acids, that means our body is going to make enough of it in order to do all the reactions or whatever he wants to do with them, to use it for the protein making or other enzyme making or whatever they want to do. So that's why these are non-essential, because your body already make them. You don't need to get them from the diet, but sometimes the people, they take a little bit more of it from diet because of some benefits. So alanine, that we talk about it, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine that we were talking about in non-essential amino acid. That means your body already making it because we need it, because it's so important for the collagen, and we don't have a collagen, so we don't have a bone, a skin, or anything like that. Proline that we were talking about, the serine, and tyrosine. And tyrosine is very also important in making the catecholamines that we're going to go through it actually in a next presentation. All right, and then now we're going to come to methionine. Okay, let's first get to the structure of the methionine. Try to draw the structure of the methionine. All right. Okay, now we're going to talk about the amino acid called methionine, one of the very important amino acids with so many clinical correlations and usage. All right, guys, now we're going to go to the amino acids called methionine. Methionine, we write it down like this. Methionine. This important amino acids. In order to draw the structure of the methionine, what we need to do, the same thing that we were doing before, the common structure, it was O, OH here, and then we have the NH group here, and then here is what actually comes up the trick. In order to understand the methionine and try to draw the structure very easily, just remember this M. Okay? Try to draw an M here. 1, 2, 3. There we go. This is the M. Okay? And what you need to do here, you just grab this and clean up a little bit. It did a lot. Okay, not a problem. So we come here and we're going to keep a little bit of the gap here. That means broken M. And over here, in this gap, we're going to put the S, which is sulfur. This is methionine. You see how easily you're going to be able to draw it when you know the tricks? The M, a broken M here, and you put the S, which is going to be sulfur here, is going to convert it to the methionine. This is the methionine, and it's very, very important amino acid. First of all, this is one of those amino acids which is like, as far as we look at over here, is going to be non-essential amino acid. It's an essential amino acid. So methionine is essential amino acid, and it's used to create the glutathione, metabolism, angiogenesis, DNA methylation, cancer, and stuff. Okay, we're going to go one by one through everything that we said right over here. How this amino acid methionine, it's making the glutathione. First of all, the glutathione is an antioxidant, very important antioxidant in our liver, that what it does actually, it's gonna clean out all these free radicals, oxygens, and peroxide, and anything that is gonna actually damage our body, liver. This is the detoxifier. Very, very important and very, very powerful detoxifier is called glutathione. And glutathione, the precursor of the glutathione is methionine. Glutathione. Glutathione, it comes from the amino acid methionine. But how this thing is going to get converted to this? Through SAM. SAM is a pathway. So SAM means S-adenosyl methionine. I'm just going to put meth over here, that instead of like methionine, we're just going to put the meth, that we don't need to put that much thing. So the methionine, it's a very important amino acid that it actually had to get to our body in order to make that glutathione. And glutathione, it's one of the most important detoxifier in the body, as far as I told you, because inside the liver, we know that everything that goes, liver unfortunately is an organ. It's a poor organ because it deals with the construction and deconstruction. That means like it creates the stuff, it makes glucose, try to convert the glucose to the glycogen and store them, is a producer and also detoxifier. So it does a lot of things in our body. But what it actually makes the liver go bad is just because of those detoxifier and so much toxicity. The toxicity is going to make the liver actually bad. So if we don't take enough methionine, what's going to happen? The liver, it's going to get damaged because of these lack of glutathione, or not to having enough glutathione in the liver. And most of the actually, even the alcohol is going to affect the glutathione. And also something very important that you guys need to know about the glutathione, this molecule is the medication called, let's get a little bit to the pharmacological aspect of the glutathione as well. So one of the most important thing about the glutathione, it's Tylenol and analgesic that we are using almost like on a daily basis. The people, they are taking it for reducing the fever, to getting out of the pain. So, and the what do you call it? The scientific name of the Tylenol is acetaminophen. which is acetaminophen. Okay, acetaminophen actually affects on the glutathione. All right, right now, what happened, this when we take too much acetaminophen, it's going to affect the glutathione level, and it's going to cause a toxicity, and the glutathione, they cannot actually fight with that much acetaminophen. That's why it's going to damage the liver. They cannot detoxify it so fast. They don't have that much like glutathione right there to do that. That's why taking too much acetaminophen is going to damage your liver. Okay, right now that we are here and we are talking about the structure of this stuff, let's see if we can even draw the structure of the amino acid, not the amino acids, I'm sorry, or how we're going to be able to draw the structure of the acetaminophen. Let's see. Right now, I'm just going to draw it and explain to you exactly how I draw it so easily. Okay. All right. And okay. You see that by heart, I could draw the structure of the acetaminophen. I bring up this thing because I want to tell you that most of these organic structure, especially from the organic chemistry, their names, I'm going to tell you exactly how their structure is. Acetyl, 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 acetyl. What is acetyl? Acetyl plus amino, amino, fen plus phenol. You see, if you combine these three together, acetyl, acetyl group plus amino group plus the amine, better to say amine group plus phenol, you're going to have acetaminophen. You see that? I'm going to individualize it a little bit with another color that you guys can understand it better. You see that right here? This is the acetyl group right here. This is the amine group. Why is NH is not NH2? Because actually it's connected to both carbons over here and here. That's it. And where's the phenol? Right here. You see how easily you're going to be able to draw the structure of the medications. And you simple. I told you guys I'm over here to simplify the medicine and also the biochemistry and organic chemistry for you guys. So you see that right here, acetyl, amino, phenol. So it's going to be acetaminophen, but they just make it brief. So this is one of the most important part of the one of the most important function of the methionine, which is so important for the body. Okay, another thing which is very, very important is DNA methylation. Angiogenesis, that means, you know what genesis, that means to making something, okay? And angio, that means like arteries, vessels, and also that means like the methionine is used to make the arteries, vessels, to make the, what I call the wall of these important structures in our body. And in the metabolism, okay, we know that how important is methionine inside the metabolism. But one of the most important things are these two things, DNA methylation and the cancer. Okay, if we want to understand these two very well, we have to understand what the DNA methylation means. DNA methylation actually means, all right, guys, we're going to talk about the DNA methylation. Why DNA methylation is important. What the methionine does. Methionine actually, it attaches an methyl group to the DNA. That means it's going to be the precursor. It's going to be precursor of doing such an important thing. And why methylation of the DNA is so important? Let me just... Okay. It's so important to have the DNA methylated. That means the methyl group to be added to. Why? Because actually DNA methylation is going to cause the repressing the synthesis phase. DNA methylation actually is going to cause the S phase or synthesis phase of some of the cells in our body to get repressed by repressing the gene on the chromosome. So that's why DNA methylation is so important. And if it doesn't happen, what's going to happen? It's going to cause the cancer, because those cells, like the muscle cells and the nerve cells, they need to grow. They need to grow, but they don't need to reproduce anymore. if they get reproduced, if they start reproducing more, if they get to the synthesis phase, that means it causing the cancer. So what it does, the DNA methylation is going to repress those genes that they are in charge of the synthesis of the cell, and start like making it like a mitosis and everything like that. So the cancerous cell actually, they don't have that mechanism. That means what happens in a cancerous cell, you don't have a DNA methylation. That's why they just go and they don't care about anything else. And if you remember in cell biology, we had like lamellipodia and philomepodia. These two elements, which are so important inside the cell, those were the cells that they were, when they were moving, and they were just like making the tissue of a certain structure, for example, liver, when they were getting closer to another structure or another organ, what they were doing, they were stopping right there. Because why? Because of those sensation. They were actually respecting the other borderline and not going forward. But unfortunately, the cancer cells, they are salvaged. They don't care. They are just like going and going from the one organ to another organ. But that's why getting enough methionine is very important for the DNA methylation. And hypomethylation is going to cause all those actually genes to start actually getting awakened and start doing the same thing that we're doing, the synthesis, the process of synthesis and making a cancer. But one thing, it's bad about the taking so much methionine, especially if the person is diagnosed with the cancer. This is very important to put him in a certain diet. Because why? Okay, here's the metabolism part. Okay, why actually the person on the cancer, diagnosed with cancer, it has to actually stay in a certain diet of the methionine. That means he has or she has not to take so much methionine, and that's going to be very harmful for him. Why? Because in our body, in order to actually, because it is essential amino acids, and you know, our body is kind of stingy. Try to reabsorb everything, try to recycle everything, try to reuptake everything. You see all these words that I'm using, reuptake, recycle, reabsorb, they are used in a different part of the body, in the different organs. For example, reabsorption, it happens in the kidney, is a large intestine. Or for example, when we say reuptake, it happened in a synaptic cleft area. And here that we are talking about the synthesis of the RNA. Actually, our body needs the methionine for synthesis of the RNA. So what happened in this case, when the cancerous cell, it's using the methionine in order to reproduce more genetic elements like RNA. So what happens, our body, because I told you is stingy, try to recycle that methionine. But that methionine recycling, it just only happen inside the cell, inside the normal cell, not the cancerous cell. So the cancerous cell, they don't have the methionine recycling in order to use it for their RNA. That's why if the person takes too much methionine, that means it's going to help those cancerous cells to reproduce more. But when it doesn't take that much of it or less of it, what's going to happen, it's going to create actually like enough for the body, for the normal cells, but it's not going to create extra for the RNA structure and RNA reproduction of the cancerous cell. This is very important. The cancerous cells, they don't have the recycling system for the methionine, but the normal human being cell, they have the methionine recycling system, which is very important. Okay, another thing, it's hypermethylation of the DNA. So we talk about the hypomethylation, that means less methylation, and now we have to talk about the hypermethylation of the DNA. Hypermethylation of the DNA due to the excess of the methionine in the body also not only is going to repress the genes that are in charge of the synthesis, S phase, and synthesis and reproducing the muscle cells and nervous cells, it also start repressing the genes that are so important.